Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today, we're going to talk about the endoplasmic reticulum and the Golgi apparatus. So the endoplasmic reticulum is a large system of folded membranes, and it accounts for over half of the membranes in some cells. There are two types of endoplasmic reticulum, rough and smooth. Rough endoplasmic reticulum we're going to call RER, and smooth will be called SER. So SER lacks ribosomes, and its functions include lipid and steroid synthesis, detoxification, especially in liver cells, and calcium ion storage. This is most notable in muscle cells as the sarcoplasmic reticulum, which we will talk about in our muscle section. Moving on to the RER, this is studded with ribosomes on the cytosolic side, and this is why it's called rough, because it has a rough texture, because there's ribosomes. There are two sites of protein synthesis in the cell, either on the free ribosomes in the cytosol, or on ribosomes bound to the RER. The proteins translated on free ribosomes are headed toward the mitochondria, nucleus, or peroxisomes. The proteins translated on the RER ribosomes can either be integrated as the plasma membrane proteins, or they can be in the membrane or the interior of the endoplasmic reticulum, Golgi apparatus, or lysosomes. And this transport happens with vesicles. So just to recap, if it's free ribosome translation, then it'll go to the mitochondria, nucleus, or peroxisomes. If it's RER ribosome translation, then it'll go to the plasma membrane, ER, Golgi apparatus, or lysosomes. Now, where a protein gets translated depends on the protein itself. All proteins start translation in the cytoplasm, but some proteins have an amino acid sequence at their end terminus called a signal sequence. This signal sequence is recognized by the signal recognition particle, which is the SRP. This binds to the ribosome. Now, the RER has SRP receptors that dock the ribosome SRP complex, and translation begins pushing the polypeptide into the ER lumen. Here, the signal peptide is removed and the protein is sent to where it needs to go. Now, secreted proteins are transported in the interior of vesicles through the Golgi to the plasma membrane, where they are released by exocytosis to the extracellular environment. Integral membrane proteins have sections of hydrophobic residues called transmembrane domains that pass through the lipid bilayer. The transmembrane domains are essentially signal sequences that are found in the interior, so not on the end terminus, and they are not removed after translation. A single polypeptide can have multiple transmembrane domains passing through a membrane. The transmembrane domains are threaded through the endoplasmic reticulum membrane during translation, and then they are transferred to the Golgi and plasma membrane, just like secreted proteins are. The RER also performs initial post-translational modifications of proteins. Now, glycosylation is associated with the Golgi, but some also occurs in the ER lumen, and this is just the addition of polysaccharides to proteins. Disulfide bonds also form in the ER lumen. And a bit more about protein transfer throughout the cell. So the default target for proteins that go through the secretory pathway is the plasma membrane. Now, if the protein going through that path wants to go somewhere else, then targeting signals are needed. Secondly, proteins that are made in the cytoplasm and need to be sent to an organelle that's not part of the secretory pathway, just like the nucleus, mitochondria, peroxisomes, need localization signals. Just to do an overview of protein transport, I'm going to give some examples. Now, if a protein's final destination is a secreted protein, then it needs a signal sequence, but it does not need localization signal or a targeting signal, and it does not have any transmembrane domains. Examples of this would be antibodies, neurotransmitters, or peptide hormones. Now, if it's a plasma membrane protein, then it will once again need a signal sequence, but it won't have a localization signal or a targeting signal, and it will have transmembrane domains. These are things like receptors and channels. If you want to go to a lysosome, you will need a signal sequence, but no localization signal, and no transmembrane domains, but you will need a targeting signal. And same for the rough ER and smooth ER. If you want to go there, you need a signal sequence, but you have no localization signal, no transmembrane domains, but you must have a targeting signal. Now, if you want to go to the cytoplasm, you don't need anything because that's where you normally are. And if you want to go to the nucleus, then once again, now you don't need a signal sequence, but now you need a localization signal. And you don't need a targeting signal, and you don't need transmembrane domains. And the same applies for mitochondria and peroxisomes. You don't need signal sequences, but you do need that localization signal. And you don't need transmembrane, you don't have transmembrane domains, and you don't need a targeting signal. And examples, so let's go through examples of all of these. Examples of plasma membrane proteins are receptors and channels, lysosomes or acid hydrolases. Rough ER proteins are enzymes that are required for protein modification. For smooth ER, it's enzymes required for lipid synthesis. For the Golgi, it's enzymes required for protein modification. And then you have glycolysis enzymes in the cytoplasm. In the nucleus, you'll have histones and DNA and RNA polymerase. In mitochondria, you'll have Krebs cycle enzymes. And in peroxisomes, you'll have catalase. So these are all just based on what their functions are. And I hope that overview made sense. Now with that, let's move on to the Golgi. So the Golgi is a series of flattened, membrane-bound sacs called cisternae. It modifies, sorts, and packages proteins and lipids received from the endoplasmic reticulum. The vesicle traffic to and from the Golgi is mostly unidirectional, and this is because sometimes proteins that are supposed to reside in the endoplasmic reticulum accidentally escape and must be returned to the endoplasmic reticulum from the Golgi. This is called retrograde traffic, and that's why it's called mostly. Otherwise, it should always be unidirectional. Now, each region of the Golgi has a different appearance and enzymes. The portion of the Golgi nearest to the RER is called the cis stack, and this receives vesicles containing proteins from the ER. The proteins from those vesicles are modified and transferred to the medial sac, which, as the name suggests, is in the middle. Here, they are further modified and sent to the trans stack, which ships modified proteins to their final destination. And some of the modifications the Golgi does is glycosylation, phosphorylation, and sulfation. And that's it for today. Next time we'll talk about vesicular transport.